All right, so we start with the first question, which says the diagram below shows a single algae which lives in fresh water. Which part of the cell labeled above traps light for photosynthesis? So what do you think is the correct answer?
It's the chloroplast. It's the chloroplast.
Chloroplast, right? Is it made of
Mm-hmm.
cellulose? Oh, is
The
made of.
chloroplast, no.
No, they're not asking about the chloroplast. They're asking for the organelle, which is organelle or part of
Which part of the?
is made of
Oh,
cellulose.
okay. Then it's cell wall, cell wall, cell wall.
Exactly. Okay. In
Yep.
the freshwater environment, water enters the algal cell. What is the name of the process by which water moves into the cells? Easy. The movement of water?
Um, Osmosis.
Osmosis, right? Give the reason why the algal cell does not burst.
It has a cell wall, which keeps the pressure.
Which can retain the turgor pressure, right? And the cell does
Yeah,
not burst.
it has a cell wall basically. Yeah.
Mhm. The algae can photosynthesize complete the word equation for photosynthesis. So water plus carbon dioxide
Carbon dioxide.
in the presence of light energy produces glucose plus oxygen.
Yeah.
Okay, the flagellum helps the cell. So this is the flagellum. Oh, they don't have the flagellum here. Oh, they do. These are the flagellum, which helps in the mobility of the cell. So the flagellum helps the cell to move through water. Scientists think that the flagellum and the light-sensitive spot work together to increase photosynthesis. Suggest how this might happen.
Um,
This is a
the flagellum makes the algae move towards the light, move
exactly because
towards the sunlight.
sunlight
Oh, okay.
can not reach all parts of the water body, right? It could reach certain parts of
Hmm.
the water body. So the
Yep.
flagellum, just like you said, that the flagellum helps the cell to move into the parts where there is enough sunlight or where there is maximum sunlight. And the light sensitive spots, they can detect that sunlight, right? And
Oh, okay.
so those spots
So
are basically
it's basically the same thing.
light.
Wait, no, they're not the same thing.
Yeah, so since they are sensitive to light, they can respond, like when it senses that when the receptor cells of that particular area, when it senses the presence of light, then the
Hmm.
flagellum, starts to aid the cell so that the cell can move through water.
Hmm. Yeah.
So the light sensitive spots basically work as receptor cells. Okay. Multicellular organisms often have complex structures such as lungs for gas exchange. Explain why single-celled organisms like algae do not need complex structures for gas exchange.
Hmm. Is it because like its cell itself has like it has a larger surface area and to volume ratio,
Okay, so
and so
first of all, since algae is one cell thick, right? Uh,
hmm
not one cell thick, but I mean, since algae is single-celled organism, um, there is the diffusion of gases. They occur through the membrane, the outer membrane. So gases can, directly diffuse in and out of the cell continuously. So there is no need for it to pass from one cell to another, right? So
hmm
that is one reason. So you could actually write it like this. Mm, You could actually,
I cannot see I cannot see it.
yeah, just a second.
All right. Okay.
Okay, so can I dictate it and would you be able to write that down? Or do you want me to
Yeah.
write it down for you? I can do that. Okay.
Yeah,
I'll
okay, that would be better.
write it down. Just a second. So first of all, single-celled organisms like algae do not need complex structures for gas exchange because of their small size and simple structure, because they allow direct exchange of gases with their surroundings, like I mentioned before. So because of their small size and single cell, they allow direct exchange of gases from their surroundings. That's one reason. And then you have that they have a relatively larger surface area to volume ratio, and thus diffusion of gases occurs at a faster rate.
Yeah.
That is one point you mentioned, right? And also, since algae has a flagellum, it helps algae to move into water body with high concentration of oxygen. They basically can move in the regions where the concentration of oxygen is high. Okay.
Yeah. Yeah, okay, we can move on.
All right, so moving on to the next question.
Completely
Photosynthesis, right? Carbon dioxide plus
water, yeah,
water, right?
with light energy makes glucose plus oxygen.
Great. Could you read this out, please? This question?
Yeah, some students investigated the effect of light intensity on the rate of photosynthesis
Mm-hmm.
in pondweed. The diagram showed the apparatus the student used: thermometer, a
Mm-hmm.
water glass beaker. The closer the lamp is to the pondweed, the more light the pondweed receives. The student placed the lamp at different distances, d from the pondweed. They counted the number of bubbles of gas released from the pondweed in 1 minute from each distance. A thermometer was placed in the glass beaker. Why was it important to use a thermometer in this investigation?
Yeah, why do you think it was important?
Is it is it to keep the temperature, like
Con
the desired temperature?
Yeah, because you want a
A constant Yeah,
unbiased
okay.
experiment, right? You don't want the temperature to vary throughout the experiment, because that would then become another factor you would have to take into consideration, right? So you want unbiased experiment, and therefore you must maintain a constant temperature, right?
To make sure it also like it isn't changing throughout.
Mm-hmm. Right.
Okay.
Okay, moving on to,
Yep.
oh, okay, you have 3 marks here. Okay, so I think it's better if you explain it a little more. So firstly, you would say that to carry out
We
an
c-
unbiased experiment so that the temperature is controlled, or the temperature is unchanged throughout the investigation, because the rate of photosynthesis, you will go for an explanation afterwards, okay? You will go on explaining that temperature can affect the rate of photosynthesis, since
mhm.
photosynthesis has some enzymes involved, right? So
Yep.
and enzymes are affected by temperature. You could say that at too low temperature, the rate of enzyme controlled reaction occurs at a very slow rate. but
And
like
then too high
temperature is too high, then the active
becomes,
side of the enzyme gets denatured, and the
yeah,
substrate molecules like water, carbon dioxide, they cannot bind, and therefore the reaction stops. Okay, so you would have to explain this because the mark here is 3 and you have enough space. I would definitely suggest you to go on explaining it. Okay.
yeah. Okay.
All right, great. So moving on to the next question. Can you please read out C for me?
The student counted the bubbles 4 times at each distance and calculated the correct mean value of the results. The table shows the student's results.
Mm-hmm.
I calculate the mean number of bubbles released per minute when the lamp was just 40 centimeters from the pondweed.
Right.
Calculate, okay, okay, wait, let me just do that. It would be 15.
The mean for
The
this,
mean would be 15, the mean number of bubbles. Yeah.
did you take this into consideration? Did you add this as
Yep,
well?
30 plus 10
Oh,
plus
you
9
you
plus
should
11.
not add this. You should not add this because this is an anomalous value. And you always omit anomalous values while doing the calculation.
You ignore them. Oh,
Yeah,
okay.
so you just add 10
Yeah,
plus
you
9
always
plus
ignore them.
11. Mm-hmm. So basically
Yeah,
the
that would
answer
be
is
10. It would be
10.
10 bubbles.
Yep. So be very
Hmm.
careful about these. And as
Yeah,
far as
all
this
right.
is concerned, you
Hmm.
would have to plot this. I would suggest if you could get a printout of this and do this, that would be much better. Okay. Uh,
Hmm.
so for now, we'll just skip it. But at home, I would suggest you get a printout of this. So moving on to question 3, which says one student concluded that the rate of photosynthesis was inversely proportional to the distance of the lamp from the plant. Does this data support the conclusion or not? Can you perhaps roughly plot this graph on your paper and see if the data actually supports it or not? Just a rough plotting so that you can guess the
Yeah, okay.
shape of the curve. Can you try to do that?
Okay. Yeah, I can do. If you can just keep it on that.
Do you want me to like go here?
No, no, you can just keep it on the thing, on the table.
Okay,
Yeah.
sure. And then I would also like you to write down the answer to question number 3 and give me a picture so that I can have a look at your answer.
Yeah, I've sent you a picture for the graph.
All right, just a sec.
We got a printout, so I was able to just do it on it.
Okay, so as the distance increases,
Oh, wait, I forgot to label the y-axis, which is basically the number of bubbles.
the number of bubbles per minute also increases, right? But like at
Mhm.
certain time, at a certain distance, sorry, it no longer increases, it falls because at that particular section, like here, the picture that you have, the picture that you send me, that particular region, the limiting factor is no longer the distance, rather some other factor is acting as a limiting factor, right?
All right. Yeah, okay.
All right, wait just a second. Let me understand one thing first. As the distance increases, the number of bubbles is decreasing. Okay, okay. So here's what, sorry, just omit what I said previously. Initially, when the distance is like, 10 centimeter,
Mm-hmm. Yep.
mm, then you see that the number of bubbles is increasing slowly, right? Like as the distance is
Yeah, yep.
increasing,
Mm-hmm.
the number of bubbles is also increasing. So that part does not support the trend, does it?
It doesn't. No, no.
But like the other part, like from approximately 15 onwards to 40, it does support the
Mm-hmm.
trend. So
Yeah,
now
yeah.
why this part doesn't support the trend is something that we will be explaining, yes. But can you please start mentioning the 15 to 40? The first line should be that, the distance 15 to 40
As
supports. Data from 15
okay,
to 40 supports and why it supports.
that as the distance increases, the rate of bubbles decreases.
Mm-hmm.
Okay.
So here's the explanation. After keeping the lamp at a certain distance, like, very close to it or whatever, it requires some
Yeah.
time for it to um, carry out the process, right? So at that very time, some other factor is acting as a limiting factor. Okay, and that is why it doesn't support the theory where it says that rate of photosynthesis was inversely proportional to the distance.
Mm-hmm.
Can you show me what you wrote after you've written it down?
Yeah, okay. Yep, I've sent it. Do I need to also say that
Yeah, you
it's
definitely should mention the part where it doesn't. So you'd say that no for 0 to 15 approximately, right? Centimeter. The data does not support theory because although as distance increases, the number of bubbles also increases. So that doesn't indicate it's inversely proportional, right?
so from
Five
15 to so from 15 to 40 centimeters,
centimeter
the
it
conclusion is correct, right? And then from
supports the theory. Yeah.
15, okay. Wait, isn't it from, okay, yeah, my bad, yeah. Yeah, so from 0 to 15, it doesn't support the theory. And from 15 to 40, it does support the theory.
Yep. So are you clear
Okay,
with the question?
yeah, that is clear now. Yeah.
You're wonderful. Could you please read this out? This is a big question. Please read it carefully.
Okay, the light intensity, temperature, and concentration of carbon dioxide are factors that affect the rate of photosynthesis.
Mm-hmm.
Scientists, scientists investigated the effects of these three factors on the rate of photosynthesis in tomato plant growing in a greenhouse. Plants growing in a greenhouse. The graph below shows the scientists' results. A farmer, a farmer, a farmer in the UK wants to grow tomatoes commercially in a greenhouse. The farmer I read about the scientists' investigation. During the growing season for tomatoes in the UK, natural daylight has an intensity higher than 30,000 lux. The farmer therefore decided to use the following conditions in his greenhouse during the day. 20 degrees, 0.1% carbon dioxide, no extra lighting. Suggest why the farmer decided to use these conditions for growing the tomatoes. You should use information from the scientist's graph in your answer.
Hmm. Okay. All right.
Okay, so it's a 4 marker.
Yeah, let me just go through the question once again.
Yeah, sure.
All right, just a second. So can we say that the farmer decided to use this condition firstly because he wanted to make maximum profit, right? Because if you look at these conditions over here, there is no
Mm-hmm.
extra lighting. So if there's if the farmer provides extra lighting, that would be providing more, I mean, like that would be accommodating more cost, right? So for
Mm-hmm. That'll be a lot more costly, yeah.
make more profit to make the process cost effective, right? And
Yeah,
then
okay,
look
that's one
at,
point to make it cost effective.
yes, and then if you look at the graph, you'll see that at 25 degrees Celsius, raising the temperature to 25 degrees Celsius makes very little difference, right? I want you to have a look at this and this. Like you can see the difference in
Is it
raise.
okay if you can explain like the graph? If you can explain the graph
Yeah, sure. Can you please
in
have a look?
detail?
Can you please have a look at this?
Mm-hmm. Yep. So that's the temperature and is the carbon dioxide concentration.
So do you see that for both the carbon dioxide concentration is the same, right? It's 0.03.
0.0.
And can you see my
Yeah.
cursor?
Mhm.
You can see my cursor, right?
Yeah, I can see it. Yeah, I can see it. Yeah.
Okay, great. So I want you to make a comparison between these two graphs. Since both the carbon dioxide, for both the graphs, the carbon dioxide concentration is the same. I just want you to forget about the carbon dioxide concentration. I just want you to look at the temperatures here because they are making the difference here, right? So do you see how raising the temperature from 20 to 25 makes a very little difference in the rate of photosynthesis? I mean, there is an increase
Mhm.
certainly,
Yeah,
but the
but
increase
it's very less.
by a
Yep.
very slight amount, right? So the raise
Mm-hmm.
in temperature does not really help the farmer in any way, right? I mean, it does
All
help
right.
him in a certain, in a slight way, but it's
Skyway.
not a
Yep.
significant profit that he's making.
Mm-hmm.
And therefore, he chooses 20 degrees Celsius because perhaps that's more cost effective, right? So
Mm-hmm.
another
Yeah.
point here would be raising the temperature to 25 degrees Celsius makes very little difference at 0.03% carbon dioxide.
Mhm.
Okay.
Yeah.
Yeah. However, if you look at 0.1% carbon dioxide concentration and let's say this one, because this two, the ones I ticked, they have the same temperature, 25 and 25 here, right? So forget about the temperature for now because they are the same for both. But just look at the graphs, how different
Yep.
they are. I mean, the increase is by a large amount, right? It makes a big difference.
Mm-hmm.
And
Yeah.
therefore, there is a significant profit. So the profit is much higher if you raise the carbon dioxide concentration. And that is why the farmer decided to go with 0.1% carbon dioxide
Carbon
instead of
dioxide.
0.0,
Mm-hmm.
right? So
Mm-hmm.
in fact, you could also
And
the values and
for
like about
mhm.
how much is it here? Wait, let me just try to read the values. I'll zoom in.
All right.
Do you see that for this, it's around 7, right? And for this, it's around 27, right? So it's like almost, uh, it's more than 3 times, right? Yeah,
Yeah.
it's more than 3 times. Okay, so can you please write these points down and just show me a quick picture of everything that I just said?
Okay.
Your answer?
All right.
And also, can you please join the next meeting?
Yeah. Okay. I'll rejoin.
Okay, thank you.